What's up guys, BIOBEAN here. So today we're going to be talking about DNA structure and how DNA is replicated during S phase. So let's get started. So DNA is made up of building blocks called nucleotides, and these nucleotides are composed of three components. The first one being a phosphate group attached to a pentose sugar, which is then attached to a nitrogenous base. And in this case, this nitrogenous base is thymine. So DNA has directionality, and it gains this directionality based on each individual nucleotide. And so the nucleotides get their directionality based on the pentose sugar carbons. So looking at the carbons within the pentose sugar, which I've labeled here, we can see I've labeled them 1 to 5. So looking at the fifth carbon, we can see that that's the carbon that's attached to that phosphate group. And so we always know the 5' end of the DNA because it's that part of the DNA that has that exposed phosphate group. So whenever we talk about the 5' end of DNA, we're always talking about that exposed phosphate group. Now, looking at the 3' carbon, so the third carbon within this pentose sugar, we see that has a free OH group or a free hydroxyl group. So this is the group that is going to interact with the next nucleotide that is going to be attached to the growing DNA chain. And so there's going to be a chemical reaction here where the H is removed and a bond is created with the next nucleotide. And it would attach, the next nucleotide would attach using its phosphate group. And so when we talk about 5' and 3' directionality in DNA, that is where we are getting it. So the 5' end is always going to have that exposed phosphate group, and because it's called 5' because it's attached to that 5' carbon on the sugar backbone. And then that 3' end is always going to be the end that has that exposed OH group, and it's called 3' because it is attached to the third carbon in that pentose sugar. So as I mentioned, that 3' OH is going to be where the next nucleotide is added. And so we have to remember that polymerase is always going to add to the 3' end of any DNA strand. So anytime DNA is being synthesized, it starts at the 5' end, and each new nucleotide is being added at that 3' end, where it is able to interact with that OH and create a linkage with the next base. So in this example, I showed you a thymine nucleotide, but what about the other ones? There are 4 different nitrogenous bases that can be added to a sugar phosphate backbone and counts as a nucleotide for DNA. So those 4 nitrogenous bases are adenine, guanine, thymine, and cytosine. And those 4 are broken up into 2 groups. So adenine and guanine are part of the purine group, and thymine and cytosine are part of the pyrimidine group. And these are grouped based on the structure of the actual molecules. So in the case of pyrimidines, they only have a single ring of carbons, whereas the purines contain 2 rings of carbon. So that is why they're grouped that way. These different bases undergo what's called base pairing. So the way that base pairing works is the nucleotide on one strand of DNA will do hydrogen bonding with the nucleotide on the opposing DNA strand. And there is only a particular pair that is ever able to happen. So for adenine, its pair is always going to be thymine. So anytime that you see an adenine on one strand, you know that the other strand will have a thymine in the corresponding place. And the same is true for guanine. Guanine is always going to be paired with cytosine. So anytime that you see a guanine on a strand, you know that the opposing strand will have a cytosine at the same place. So here we're looking at 2 DNA strands, which together make up that DNA helix that we're very familiar with when we think of DNA. And so, as I said, the base pairing rules always hold true. So anytime that you see an adenine on one strand, you're going to see a thymine on the corresponding place. And these 2 strands are said to be anti-parallel. So what that means is that they run in opposite directions. So if you remember, I said that all DNA strands have directionality, and that directionality is the 5' to 3' direction. And that 5' and 3' directionality is given based on the 5' carbon, which is attached to a phosphate group, and the 3' carbon from the pentose sugar, which has a free OH or hydroxyl group. And so these 2 DNA strands, which come together to form a helix, run in opposite directions. So the top strand here is in the 5' to 3' direction, whereas the bottom strand is running in the 3' to 5' direction. So while they're parallel to each other, they're termed anti-parallel because they're running in opposite directions. So they're not running together in the same way. So now let's get a little bit into how DNA is replicated. All right, so first I'm going to give you a brief overview of how replication is actually done, and then we're going to get into an experiment of how we came to find out how DNA did this. DNA replication is said to be done in a semi-conservative manner, so let's get into what that means. When DNA is replicated, the two strands of a helix is pulled apart. Once the strands are pulled apart, a polymerase runs along each strand, and it reads what base is there. And as we know, the base pairing rules mean that anytime we see a T, that polymerase is automatically going to know to make a new strand that has an A. Once the polymerase has finished copying down all of the new strand, we are then left with two sets of DNA helices that contain the same information as the original. Now, when we actually go to do mitosis, these are going to segregate into two separate cells. The two daughter cells that result at the end of mitosis now carry the same genetic information as the original cell, but now each cell gets a single strand that was from the original cell and a new strand that was newly synthesized during that S phase before we entered into mitosis and separated into two cells. So that is why DNA replication is said to be semi-conservative, because a strand of the DNA is conserved from the original cell, but one of the strands contains entirely newly synthesized DNA. So it is semi-conservative because we conserve some pieces of the DNA, but some of them come from a new round of replication. So now we're going to get into the experiment that was done in order to discover that DNA replication was done in a semi-conservative manner. It's a little bit complicated, so I'm going to try and break it down as simple as I can, so please bear with me. So there are these two guys, Meselson and Stahl, and they hypothesized that there were three possible options for the way that DNA replication was happening. So their first hypothesized way of DNA replication was conservative replication. So they hypothesized that conservative replication would occur where the two DNA strands are pulled apart and DNA replication happens to create those two new strands. But then the two old strands go back together and form a DNA helix, and the two new strands go back together and form a DNA helix. So that's depicted here. Anytime you see black DNA, we're calling that old. Anytime you're seeing red DNA, we're calling that new DNA. So at the end of conservative replication, you would get two DNA helices, one with entirely old DNA from the cell and one from totally newly synthesized DNA. So the second option that they were trying to hypothesize about was semi-conservative replication. So in semi-conservative replication, you have the two DNA strands pull apart, replication happens to create those two new strands, and then they remain together. So you have one strand of a helix having old DNA and the other strand of the helix having new DNA. And it created two DNA helices that have that mix of one old strand and one new strand. So that's depicted here. And the third option that they hypothesized about was dispersive replication. So in dispersive replication, somehow the two strands would pull apart and then certain chunks would be old DNA and certain chunks would be new DNA, but across both strands. So logically, dispersives kind of seem really difficult. We could not really think of a mechanism that would have happened, but in science, we don't know until we actually do the experiment. So they ran with the dispersive theory. So they were trying to design an experiment that would be able to tell between these three different types of replication to determine what type of DNA replication was actually happening. So here's what they did. So what these guys did is they took a cell and they put it into a beaker containing heavy nitrogen. So this was able to be detected because it's radioactive. So by putting the cells inside heavy nitrogen, it then marked all of the DNA with heavy nitrogen. So any DNA that was already present within the cells were going to be marked with that heavy nitrogen. So we're trying to mark that old strand of DNA so we can follow it through the different replications to see what type of strands we get. Because we're trying to determine between these three options for the type of replication that cells do. So once all of the DNA had been marked by the heavy nitrogen, they then placed the cells into light nitrogen. So now anytime that DNA is going to be incorporated, it is going to be marked with that light nitrogen. So now we've marked all of our new DNA with light nitrogen. They then allowed the cells to undergo one round of replication. So now we're going to go over the different predictions that they would have seen in the DNA based on what type of replication was happening. So they allowed the cells to replicate in that light nitrogen beaker, and then they pulled the cells out and then isolated the DNA and put them into a beaker and centrifuge them with cesium chloride. So what that does is that actually separates the DNA based on weight. So what we would predict to see if DNA was being replicated using conservative replication, we would expect to see two independent bands of DNA. One independent band being the heavy DNA, and that's going to be our old chain of DNA, and one band of light DNA that is going to be our entire helix made of new DNA. What we predict to see if there was semi-conservative replication is there would be an intermediate band. Because each DNA helix contains both heavy and light nitrogen, because it's containing one strand of new DNA and one strand of old DNA, we would expect to see that intermediate band. So anytime we have that DNA helix mixed with old and new DNA, it is going to show us an intermediate band of light and heavy nitrogen. And then for dispersive replication, we would also expect to see the same thing as semi-conservative replication. So even though there are a mix of new and old DNA on both strands, because both strands contain old and new DNA, we're going to get a mix of light and heavy nitrogen. So they needed to do a second round of replication in order to actually differentiate between semi-conservative and dispersive replication. Because if we only did one round of replication, the semi-conservative and the dispersive would give us the same result. So they allowed the cells to sit in the beaker containing the light nitrogen for 2 rounds of replication and then isolated the DNA. After 2 rounds of replication, we would expect the conservative result to remain the same. So if replication is done using conservative replication, we would expect to still see after 2 rounds of replication, one band of heavy nitrogen, because there's always going to be 2 strands in a DNA helix that's containing the old DNA, and one band of light nitrogen, because there's always going to be a DNA helix containing entirely new DNA. So with conservative replication, that is always going to be the result. Now with 2 rounds of replication, with dispersive, we would now expect to see the same result as we would have seen within 1 round of replication. So with dispersive replication, you are always going to see a mix of old and new DNA on both strands. And so therefore, you're always going to see that intermediate mix of heavy and light DNA. And so the change really starts to emerge with semi-conservative replication. So after 2 rounds of replication with semi-conservative, we now, instead of expecting to see only one band that contains that mix of heavy and light nitrogen, because each helix contains one strand of new DNA and one strand of old DNA, after 2 rounds of replication, we are now going to expect to see one intermediate band with heavy and light nitrogen, and one band with entirely light nitrogen. So let's explain why semi-conservative after 2 rounds of replication is going to show that different result. So with semi-conservative replication, we start with our 2 original strands of DNA. And so those are represented as the 2 black strands of DNA. Those are our old strands of DNA. After 1 round of replication, those 2 old strands are going to separate apart, and a new strand is going to be synthesized for each of the old strands. So now we've created 2 DNA helices with 1 old strand and 1 new strand. And so that was why after 1 round of replication in the Meselson and Stahl experiment, we saw a test tube that had that intermediate weight of nitrogen. And that was because each strand contained an old DNA, which was marked with that heavy nitrogen, and the other strand was marked with that light nitrogen because it was new DNA. So now, after a 2nd round of replication, each of these strands pulls apart and is going to make a new helix. And so we go from having only 2 sets of DNA helices to 4 sets of DNA helices. So in the case of having now a 2nd round of replication, each of these strands is going to separate out and have a new strand synthesized with it. So the 2 old original strands of DNA are going to persist, and they're going to be into 2 sets of cells. So here I've marked it as cell number 1 and cell number 4. And so those are going to receive that old original copy of DNA with a newly synthesized strand of DNA. The other two DNA helices that are created after the second round of replication contain entirely new DNA. That is because the new strands of DNA that were synthesized in round 1 are now going to act as a template for the new strands in round 2. And so 2 sets of helices are going to contain entirely new DNA. And so that is why in the Meselson and Stahl experiment, after 2 rounds of replication, if semi-conservative replication is being done, we will see a test tube that contains that intermediate band of nitrogen containing heavy and light nitrogen. So that would be representing 1 and 4 here. And we would also see that band containing entirely light nitrogen coming from those entirely new strands of DNA that were created. And that would be what would be represented in helices 2 and 3. So, surprise, surprise, when they actually conducted the experiment, they discovered that replication was done in a semi-conservative manner. So when they looked at the test tube after 2 rounds of replication, they saw this. They saw that intermediate band containing the heavy and light nitrogen, representing cells 1 and 4 here. And they also saw that light band of nitrogen representing helices 2 and 3. And so that was how we concluded that DNA replication is done in a semi-conservative manner. All right, so we just went over how DNA replication is done in a semi-conservative manner. So now we're going to go over how replication actually takes place and the players that actually help it happen. So the cell has passed through G1 phase of the cell cycle and has now entered S phase, so the synthesis phase. So now that is when replication actually happens. So we have our DNA helices and we somehow need to get in between all these wiggles in order to actually replicate and create a new strand. So the first thing that needs to happen is the DNA needs to be unwound. So it's all wrapped up in the DNA helix and none of the proteins can really get access to it in order to copy it. So the first thing that we need to do is have a protein called helicase unwind the DNA. So DNA helicase unwinds the DNA. But because of base pairing, the DNA very quickly wants to go back to being wound up because that's what the attraction makes it want to do. And so there are special proteins called single-stranded binding proteins that bind along the unwound DNA to prevent it from going back into a helix shape. Now the single-stranded binding proteins have attached to the ends of the DNA to prevent them from going back together into that helix shape. So now we have another problem. As helicase is pulling apart the helix to create our 2 strands of DNA to allow for replication, there now is a very twisty problem. So if you've ever taken a string that's wound together and you try and pull it apart, you'll notice that it creates a really big knot. It gets very, very wound at the bottom part of where you're trying to pull apart from. So in order to solve this problem, there is a special protein. This protein is called topoisomerase. Don't come at me for butchering that. It's a really weird word. So this protein goes and it binds to the other end of where helicase is and prevents that supercoiling from happening. So this topoisomerase binds to the other end of where the DNA helix is being unwound and it prevents that supercoiling from happening. All right, I've officially run out of colors, so I'm going to start reusing colors, so bear with me with that. So now the DNA strand needs to actually be synthesized. So there's kind of a little quirky issue in that there needs to be DNA already present in order for a strand to be extended. So there somehow needs to be something laid down for the polymerase to come and actually extend that new strand. So that is solved by something called an RNA primer. So an RNA primer is what it sounds like. It is RNA that is acted to prime the polymerase to allow it to then extend a strand. So a protein called primase, named after its function, so it lays down RNA primers, and it puts down a small piece of RNA. And this RNA is going to be complementary to the DNA strand that it's being copied from. Something you have to remember that is unique to RNA is that instead of having the nitrogenous base thymine, it contains the nitrogenous base uracil. So it has all of the other same nitrogenous bases. It has an A, it has a G, it has a C. But for RNA, instead of having that T, it has a U. So we'll always know where the RNA primers are is because in place of T's, we're going to see U's. So anytime you see a U during replication, you know that that is the RNA primer that was laid down by primase. So this is done because for some reason, primase is able to add nucleotide bases down without any strand already existing there. Whereas for some reason with DNA polymerase, that's not possible. So this primase lays down that RNA primer to allow polymerase to come and extend that using DNA. All right, so now that we have our RNA primer laid down by primase, DNA polymerase 3 is now going to come in and it is going to now add DNA nucleotides. So here we have DNA polymerase 3. I think there's maybe 4. So this in particular is DNA polymerase 3, and that is the main polymerase that is going to extend DNA from those RNA primers. So it comes in and from a floating pool of nucleotides, it adds in the corresponding base. And we have to remember that it's going to add the complementary base. So it's always going to follow those base pairing rules. So if on the template strand, it has a T, then DNA polymerase knows that in the new strand, it is going to place an A, because it is always going to follow that base pairing rules. But DNA polymerase needs a little help. So it's not super good at staying on the DNA strand. So it has an accompanying protein called a sliding clamp that helps it stay attached to the DNA that it's making. And then the DNA polymerase moves in the 5' to 3' direction to create new DNA. So it's going to be moving in this direction as it synthesizes the new DNA. So now DNA polymerase has done its thing, it's run along the DNA, and it synthesized the new strand of DNA. But now we have a problem in that we have the RNA primers laid down. And our DNA consists only of DNA, so we need a way to remove these RNA primers. This is done by DNA polymerase 1. So I told you there were multiple different polymerases. So polymerase 3 is the main polymerase that actually synthesizes the DNA. And then DNA polymerase 1 is going to be the polymerase that actually chews up those RNA primers, so it removes those RNA primers, and it lays down DNA in its place. And it's able to do this because it has the already created DNA strand to run along. So now our DNA polymerase 1 came by, it removed those RNA primers, and it put down that new DNA in its place. But now we have the problem of there's a nick in the phosphate sugar backbone of the DNA. Because they were made in 2 separate chunks, that sugar phosphate backbone between the 2 chunks need to be attached together. That is done by DNA ligase. So our DNA ligase is going to come along, it's going to detect the nick, and is going to ligate the 2 strands together. So DNA ligase ligates 2 different strands together to create one cohesive strand. So those are the general players involved within replication. So now we're going to get a little bit into the more nitty-gritty parts of replication. So here we have a piece of unwound DNA helix. So this has just been unwound by helicase, and now we have what we call a replication bubble. So a replication bubble arises when 2 helicases in opposite directions move apart to create an opening of linear DNA so that the DNA can be replicated. So 2 replication forks is what they're called. So when the DNA helicase unwinds the DNA, that is called a replication fork. So a replication fork in either direction generates a replication bubble. So we have replication occurring in both directions of this bubble. So we can kind of draw a dotted line down the middle of this bubble, and replication is going to happen in the either direction of that line. So I've divided our replication bubble in half, which is now going to show us that replication is going to happen on either side of this replication bubble. But we have to remember that polymerase always synthesizes DNA in the 5' to 3' direction. So let's put some directionality on this replication bubble. So this top strand here is in the 5' to 3' direction. So if we cut this strand in half, we would say that to the left of the cut is going to be the 3' end, because if that was all that existed in the DNA, This would be the 5' end, and that would be the 3' end. And then the right side of that replication bubble, I marked as the 5' end, because if that was the only piece of DNA present, if this is the 3' end, we know that this must be the 5' end. And so the same thing applies with the bottom strand. So this one, because the DNA strands are anti-parallel, was running 3' to 5'. So when I cut our replication bubble in half, the left side of the bubble is going to be our 5' end, because if that was the only piece of DNA present, if this was the 3' end, then this must be the 5' end. And so then the same thing is true for the right side of that bottom strand. That is going to be our 3' end, because the very right side is the 5' end, and so that must make this the 3' end, because DNA always has that 5' to 3' directionality. And so we always have to remember also that DNA polymerase can only synthesize DNA in the 5' to 3' direction. So looking at our replication bubble, let's look at the different directions that DNA polymerase would have to move in order to replicate the DNA present. So looking at the directionality of our template strand, we can then infer the direction that DNA polymerase is going to move, because it moves in that 5' to 3' direction following that template strand. So for the top strand of the replication bubble, the left side of the replication bubble has the 5' to 3' directionality with the 3' towards the middle of the replication bubble. Therefore, when DNA polymerase moves, it is going to move towards the middle of the replication bubble. So we call this portion of the strand the lagging strand. And then moving on to the right side of our replication bubble, we see that the middle is the 5' and towards the end is the 3'. So our DNA polymerase is going to move from the middle towards the end of the replication bubble, towards that replication fork. And so this strand is called the leading strand. So this also applies to our bottom strand. So on the very left side of the replication fork at the bottom, it is moving in the 5' to 3' direction from the middle towards the end. So this is again called our leading strand. So anytime the middle is the 5' and it's moving towards the replication fork, which is the 3', that is called the leading strand. And then anytime the middle of the replication bubble is the 3' end and it moves towards the 5' end, that is the lagging strand. So the reason that the 5' to 3' portion of the replication bubble is called the leading strand is because it is much simpler for DNA polymerase to replicate this portion of the strand. This is because primase is able to lay down the RNA primer at the very beginning of this portion of the leading strand. And then DNA polymerase is able to come and slide along that strand and move towards that 3' end until it hits the end of the strand. But now when we look to the lagging strand, because it has to move from the 5' to 3' direction, it has to move from the edge of the replication fork towards the middle of the replication bubble. And so this forces the DNA to be made in fragments. We call these fragments Okazaki fragments. I'm assuming by the guy that found them, so we call them Okazaki fragments. So the way that replication works on the lagging strand is that RNA primer is laid down at the 5' end of the exposed replication bubble. Then DNA polymerase comes, attaches to that RNA primer, and it extends towards that 3' end, which is located at the middle of our replication bubble. But now it has the issue of hitting the end of this portion of the strand. So DNA replication is happening on all 4 sides of this replication bubble simultaneously. So because this side is our leading strand, we already would have had RNA polymerase put down RNA primer and have that DNA be extended. So when it comes to replicating the first Okazaki fragment on the lagging strand, it is going to reach a point where it hits already synthesized DNA that was made on the leading strand. So the way that this is solved is that DNA polymerase makes the first Okazaki fragment until it hits the already synthesized DNA. Then that polymerase falls off and a new primer is laid down further down towards that 5' end, because, as I said, all of this is happening simultaneously. So helicase is unwinding more DNA as DNA polymerase is replicating the already unwound DNA. So now that helicase has exposed more of this lagging strand, another primer can be laid down on that 5' end, and then DNA polymerase is going to come and replicate the DNA, moving towards that first Okazaki fragment. And so it is going to move in that 5' to 3' direction until it hits the first Okazaki fragment that it made, and then it is going to fall off and restart the process all over again. A little bit further down, there's going to be a bit more exposed DNA, another primer is going to be laid down, and then DNA polymerase is going to move in that 5' to 3' direction until it hits that second Okazaki fragment. And it continues to replicate in this way on both of the lagging strands that are present within this replication bubble. So you'll always be able to tell which is the leading and which is the lagging strand based on the directionality of the strands given to you. So don't try to memorize the left and right, top, bottom. Always try to remember it based on the directionality given to you, because they like to flip around what directionality they give to you so that you can't just simply memorize that the left is the leading and the right is the lagging. So always make sure when you're working with questions like this to break apart your replication bubble in half, and then give the directionality within the middle of the bubble as if there was no DNA present on the other side. So that'll be an easy way to give you the directionality. So if I have this 5' end, I know that the middle portion on the left side is going to be the 3' end, and so on. So now that we got into the very nitty-gritty of how replication happens, let's go a little more brief overview of how replication happens in prokaryotes versus eukaryotes. So in prokaryotes, they have a circular chromosome. So there is only a single replication bubble that occurs at the origin of replication. So the origin of replication, or ORI, is a particular sequence within the DNA that is recognized as the start site of replication. So that is where the replication bubble is going to begin, and then the replication is going to occur along either side until the full circular chromosome has been replicated. So in prokaryotes, they only have that one replication bubble. Whereas eukaryotes have a linear chromosome. And so eukaryotes have multiple origins of replication, or multiple replication bubbles. And these are all replicated simultaneously until they reach each other and then fall off and go find a new spot to go and replicate. So as I just mentioned, eukaryotes have that linear chromosome. And so there reaches a point where the polymerases will actually fall off the end of the chromosome after they finish replicating it. And so for the leading strand, that's no problem. Up here on our leading strand, which is moving from the 5' to 3' direction, with the 3' direction being the end of the chromosome, the RNA primer is laid down on that 5' end, and the DNA polymerase replicates until it quite literally falls off the end of the chromosome. So the issue comes about when we're trying to replicate the very end of the lagging strand. So on the lagging strand, we've just come to the end of the chromosome, and primase has laid down the last RNA primer, and then polymerase comes and extends that last piece of DNA. But now we have an issue. DNA polymerase 1 cannot come here and remove this RNA primer and lay down new primer because there's not enough room. So cells have solved for this issue by creating a protein called telomerase. So what telomerase does is it comes in with its own piece of RNA template, and it is able to synthesize a new piece of DNA onto the old template strand of the lagging strand. So now the polymerase is able to come and extend that portion of the overhang that was just created by telomerase. While this process will still leave a small piece of overhang where the DNA cannot be replicated on that lagging strand, it was additional DNA that was added by that telomerase right there. So that piece of DNA is not very important. It was added particularly by telomerase so that it could be unneeded DNA. So even though a little portion of it isn't going to be replicated, that doesn't matter because it's just nonsense DNA that was added on by telomerase. It's just a random sequence of DNA. So this telomerase protein was actually named because the end of every chromosome is called a telomere. And so what the telomeres are is a portion of DNA at the end of every linear chromosome that does not code for anything that we need to function. So it is a long portion of nonsense DNA that exists purely because of the fact that when replication occurs, there is going to be a portion of DNA that is left unreplicated. And unfortunately, in humans, we no longer have telomerase active in our bodies. So we are not able to continue to extend these telomeres to continue to add on more nonsense DNA as replications occur. So because we do not have telomerase active in our cells, every time that we do a round of replication, there is a portion of DNA that cannot be replicated. And slowly, every replication, a little bit of that telomere is going to be lost. We actually believe that this is what contributes to aging in humans, is the fact that we do not have active telomerase. And so therefore, we are slowly getting a smaller and smaller telomere after every replication. So by the time we get to be 70 or 80 years old, we have already run through our entire telomere. So we've already removed all the little pieces of nonsense DNA, and then we begin to remove needed DNA during every round of replication on that lagging strand. And we believe that this is what contributes to aging. So there's a little bit more to that story, and we're going to get into that in the next video, so stay tuned. So this concludes our video on DNA replication. If you have any questions about this or any other biology content, please visit biologybean.com where you can access tutoring by yours truly. Please don't forget to comment, like, and subscribe.